Just give me a minute. I'll just be back. I'll share my screen with you. I'll be taking your SEM 6 animal biotechnology and immunology classes. So I'll speak a little loudly so that you'll be able to hear me. So let us just see the basic concepts of immune system. I will just share my screen with you. Just give me 1 minute. So when it comes to the immunology and immune systems, basically, we get to see that we have different kinds of organs inside our immune system, which will protect us from different issues, whatever arise. So there is a particular system which will help us different kinds of environment that we live in, and especially the types of microorganisms or anything that comes over which is foreign, we are protected by the immune system. So when it comes to, we have different kinds of organs that are there. We have the primary lymphoid organs and the secondary lymphoid organs. Primary lymphoid organs are your bone marrow, thymus, and whereas the secondary lymphoid organs, glucose associated, blood associated, in the lymphatic system, we get to see that in the plasma from the blood, it gets into the tissues. Basically, the interstitial tissues gets into the lymph. So you have a lymphatic system that goes along with your arterial system. So it is going to take place in one direction only. Lymph nodes act as filters for antigens. That is there. I apologize for the sound today. I cannot really help, and there's some function going on next to me. So please adjust with the sound for today. So we have something called as the lymph nodes, like I told you, and it helps in filtration of particles, microorganisms, exposure, and this helps in whatever microorganisms or foreign particles are there, they are going to be removed from the blood vascular system into the lymphatic system and out. So activation of B lymphocytes takes place. Initiation or starting of the immune response takes place in the lymph nodes. So the different types of cells that are involved in the lymphatic system or the lymph node are the lymphoid cells, you have macrophages, and they're all antigen processing, antigen presenting cells as such. And then you have vascular endothelial cells and fibroblasts. They also form the supporting framework that is present. So this is how a lymph node looks. So you again have a medulla present and cortex present over here. And it is completely filled with a mesh-like system, so that there's a lot of going into and out of it, the lymph node. So you have the efferent lymph vessels and the efferent. Efferent from the outside or from the different parts in the lymph node, the lymph is being part of or getting the circulation. Then lymph vessel. And all of this simultaneously along with your blood capillaries. So it is a mesh-like system, very intricate, present. So this is how a lymphatic system is. Now we have something called as the spleen. It's the largest accumulation of the lymphatic tissue that takes place over here. What happens in the spleen is, we know that spleen is the site and the graveyard of arteries. So we know that. So So this is where all the aged or diseased blood cells are erythrocytes are going to be destroyed. It is where activation of lymphocytes takes place. So it's a very important organ that is present inside the body. And made of this lymphoid tissue is known as white pulp, and this is also be active. And this is the red pulp is actually basically where your old RBCs are removed or are filtered through. That's why it is known as the red pulp. So this is how the spleen looks like, red and white pulp passage. This is the spleen capsule form. And what are the functions? Of course, like I told you, the cells are present inside it. And then you have a germinal cell, and then this is where your red pulp is present. Then we have something known as your mucosa-associated lymphoid tissue, which is very important in our immune system as well. And we also have your gut-associated lymphoid tissue. So then these include your tonsils, adenoids that are present in the nose, behind in the ears part, so you have adenoids over here basically, and you have Peyer's patches which are present inside the intestine, and then aggregates in the appendix and so what is going to happen? Small lymphoid aggregates in each phase of gut. All of this help in seeing that you do not have different kinds of tell whenever there is antigen, this particular system, lymphoid tissue, is going to protect from it. And then your mucosal associated lymphoid tissue mold is completely consisting of your mucus layers. Wherever we have mucus at the body, right from the lining of the nostrils, to the gut, to the esophagus, to all the lung that you mucus layers. Mucus, so that like that just or starts collecting all the dust particles most in fact and stops what do you call it pathogens of and of course are also there or in fact this is how it looks like full of internal lumen and it's completely folded. Right. So this is how Payas patch looks like. Give me one moment. I want to show one point if possible, and then we'll go for the teaching cell present. That said, I'll stop my video and right. So like cells of the immune system, we have different kinds. So it identifies processes, skills, factors who must practice. Immune means exam. So body system consisting of organs, tissues, cells, and products, which helps us differentiating between the self and the non-self. Immunity basic classification, if you see, innate immunity is we get the immunity by birth. Adaptive immunity sense that natural as well as artificial. Natural is after either we get it through our maternal, that is the mother, IgG is one example. It is by infection. So we people who are right. So natural is passive and active. Artificial is your vaccine, for example. So cell's body history is right from 1798, Edward Jenner smallpox vaccination was started, and then in 1975 was when a stepping stone basically, wherein you had what do you call it, the Kohler and Milston getting in monoclonal antibodies. Where are they? That is it all over the body. I'm not going to go into detail. We've just seen it right now. So primary lymphatic organs and secondary lymphatic organs. And we all these can be at least, we have a basic protection given by our body, viruses, carcinogen, pollution, germs, toxins, all of them, in fact. What are invaders? This bacteria, fungi, parasites. Yes, with foreign particles. Yes, if you see a small thing which is there embedded inside your system, that is also affected as a foreign entity, and that is to consideration, and your immune cells will We have humoral as well as cellular responses basically, and that comes under innate immunity and acquired immunity. In acquired immunity, we have humoral wherein antibodies are produced, and cellular immune response wherein you have the T cells, that is your thymus cells, CD4 cells, CD8 cells, and others that are being released. We have the seen bursa, and in organisms such as we have bone marrow, thymus, but in the animals, so you have something called as the bursa of Fabricius. That is why we have something called as the B cells. So this is where you have the, what do you call it, maturation of the cells, B cells taking place in the birds basically, but it is absent in human beings. So these are the organs of immune system. Lymph node, like I already told you, efferent lymph node, and the outgoing, that is the efferent lymph node. This is how the lymph node is present. And we have the lymphatic vessels that are present. Development is through your lymphoid precursor, and you have your pluripotent cells giving rise to myeloid precursors and lymphoid precursors. The myeloid precursors will be what do you call it, cells, and then your macrophages. Lymphoid precursors will give NK cells, and then your CD4, CD8 cells. And from the myeloid precursor itself, you have the erythroid cells and the glioid cells coming up from. So to protect us, we need a very important factor that we have to distinguish between self and non-self. That is where we have an issue wherein certain autoimmune disorders wherein have a possibility that the cells cannot distinguish between the self and the non-self. So the cells are understood as foreign cells, and that is when we have an invasion against our own cells. This is what happens in the case of autoimmune diseases such as rheumatoid arthritis, then arthritis lung or farmer's lung, in fact, and then you have even systemic lupus erythematosus, type 3 type of hypersensitivity. So our self cells are also recognized as foreign, and antibodies are released against it. So we have to take care of that part. And anyway, when it comes to infection and immunity balance, you just see that diseases, basically infection, how much of virulence is there and how much amount of immunity is there. So what is the beneficial part of the immune system? We are being protected. And what is happening is altered cells, whatever they are there, basically, are being first killed or eliminated. But we have a collateral damage because of this. So what is it? Inflammation, pain, reddening of the skin, and of course, the different kinds of cells or immune cells are being activated. So because of which, sometimes we develop hypersensitivity or autoimmunity as such. So overview, like I told you, immune system has innate, adaptive, cellular. This is how the lymphocyte origins are present. And the functional basis has adaptive or specific immune system. And host also has certain barriers. What are they? Anatomical barriers, which include your mechanical, chemical, and biological. Whereas humoral components is where your humoral cells are are activated. That is your complementary system. Your blood clotting system is going to be activated, as well as the cytokines are going to be released. So first line of defense always is going to be a skin. Skin is the most important barrier that is present. That you have your tears. Tears have an enzyme called lysozyme, which helps us from different kinds of bacteria that comes in. Up to a certain extent. Respiratory tract. See, for the basic part that you have a gullet over here, and you have a windpipe that leads to the trachea, and you have a small epiglottis, which actually helps in seeing that you do not have anything passing into your windpipe. As soon as something, you have water, it goes in the wrong direction into the windpipe, you start coughing out. It is a reaction that is coming. It is the first line of defense that is being acted upon. So you see your skin, epithelia, different kinds of neutrophils, macrophages are your first line of defense. In your urinogenital tract also, your urine pH, lysozyme, and lactic acid that is present, gut bacteria present are all going to help. And at the same time, you have your skin, sweat, tears, which has lysozyme, like I told you. Saliva, salivary lysozyme is there. Salivary amylases are there, which have a different, function altogether. Mucus traps pathogens. When you sneeze, it comes out washed away. All the chemicals get destroyed because of it. Stomach acid destroys pathogens. pH is 1 to 1.5. So that's what. From the nostrils to the inside the input. And as you say, the first line of defense helps us a lot in the first place. And see, you can bacteria, viruses, innate response is going to be higher. Then afterwards, if this physical barrier is not going to be helpful, that is when your cells of immune system, that is your T, B, cytokines or memory cells are going to be activated. And then of course, we have an inflammatory response everywhere, and you have the histamines and the complementary system that gets activated. So histamines, what does it do? It dilates the local blood vessels. So, and then what happens when that takes place? It increases the blood capillary in that particular area. So what is going to happen because of that? You have an inflammation. You have swelling in that area, pain in that area because of different kinds of pain. Prostaglandins are released. And then because of which you have swelling. So this will make that area where there is inflammation present hot. So, and this hotness is not going to be favorable for different kinds of microorganisms. It also helps in calling up your WBCs and raising the metabolism of the cells surrounding around that particular site. Because of which you have unfavorable circumstances that are being developed over there, because of which there is a chance for the pathogen to be dead. And then you also have the complementary system that is being released, because of which chemotaxis takes place, recruitment of WBCs is taking place. Of course, again, inflammatory response is the side effect. Organs, like I told you, primary, secondary lymphoid organs are present, adenoids, tonsils are present, and then different kinds of interleukins, T cells, everything that's present over here. Thymus, as I told you, it's there. Anatomy is how it is. We have already seen this, so I'm not going to go into details as such. And histology, we have seen just this in a little while earlier. Cells of the immune system, development, how it is taking place right from your bone marrow. You have the erythroid and the myeloid precursors coming. Before that, you have the pluripotent cells, which will give rise to different kinds of precursor cells. Then later we have your T cells and B cells, which are going to be activated. So when it comes to the components of the immune system, you have the lymphocytes, phagocytes, granulocytes, others. Lymphocytes are your T and B lymphocytes. And then you have your large granular lymphocytes. Phagocytic natured cells are your mononuclear phagocytes, neutrophils and eosinophils. Granulocytes are your basophils and mast cells. And others include your platelets. So these are involved in your as inflammatory mediators. These form the antibodies, these are the cytokines, and this is the complementary system. So finally, all of these are soluble mediators in the immune system. All of these are your lymphoid cells, small T or B, blast cells and plasma cells that are there. These are the null cells. And your natural killer cells, basically, example is your CD56 that is present. And you have mononuclear phagocytes present in the blood, basically increases the phagocytic ability, and it secretes different kinds of cytokines and hydrolytic enzymes. So based on the tissue that they are present, they have different names. Example is when they are present in the brain, they are known as microglial cells. If they are present in the liver, they are known as your Kupffer cells. They are present in the lungs, they are known as your alveolar cells. So how do they get activated? By either cytokines, for example, gamma interferons, or different kinds of phagocytic action. They are going to be presented to your major histocompatibility complex 2 that is present. We will come to that in some time later. So we have two kinds of complexes, MHC1 and MHC2. So these are the monocytes that are present. This is the macrophages. It has some kind of like pseudopodia in nature, which helps in engulfing the particular different kinds of pathogens present. So there are also effective antigen presenting cells. Basically, see, this is how a bacteria is going to be engulfed by the macrophage, goes inside the lysosome, helps in forming a phagolysosome to have a class 2 NHC compatibility complex which is present, which is going to destroy this particular bacteria and releases the degraded material. We have something called as the dendritic cells. These dendritic cells are present, in certain places. Where are they present? In the Langerhans found in the skin, and as circulating dendritic cells in the myeloid and plasmacytoid positions. So what do? They help in presenting the antigen as it is, meaning it is an antigen presenting cell. Can even realize that it is a non-self cell and attack cancer. Tumor linked to a cytokine forms a complex in the dendritic cell. The tumor antigen comes and activates the T cells, because of which the cancer cells also are attacked by your T cells. So these are your dendritic cells, and the pathways. See here, how is it developed? Hematopoietic stem cells, common myeloid progenitor, common lymphoid progenitor cells. Myeloid gives rise to Langerhans, interstitial, and monocytes. Monocytes gets developed into your myeloid dendritic cells. Common lymphoid progenitor develops into lymphoid dendritic cells. Now we have the mucosal associated lymphoid tissue, like I told you. This is how it is present, known as the Peyer's patches or lamina propria. And through this, we have different kinds of pathogens that are being attacked by when they are exposed into the intestinal region. So immunity has both innate and acquired immunity, which is again developed into tumoral or cellular response based on the kind of cells that are being released. Innate immunity is wherein, when you have pathogenic infection that is taking place, and you can see skin, ciliated cells, lysozyme, coughing, and vomiting is how it is going to take place. First line of defense. Second line of defense is when a blood clot is formed, histamines are going to come, cytokines are going to come, and leukocytes are going to come and activate and remove the pathogen from the blood. And it is not specific, and there is no immunological memory. That is wherein your humoral cells are not being activated. Your memory cells will not remember that this has happened earlier. Now we have, if you have been infected with chickenpox when you were a young child, you have that particular immunity throughout your life. That is because your memory cells, even when you are exposed to chickenpox later in your life, it is going to come and destroy those pathogens. But it is not going to happen every time. So whenever there is this innate immunity being triggered, you do not have an memory that is there. So it is an immediate response with no specific recognition as such. Only neutrophils are released. So in the adaptive, you have, of course, cytotoxic and antibodies that are being released, and it will take almost 7 to 10 days for these antibodies to kick in. So, like I told you, basic, innate, and adaptive, cellular, and humoral are being released. You have a time lag in the case of adaptive immunity, whereas no time lag in the case of innate immunity. Again, antigen specific, no antigen specific. So you see a pathogen, it just comes and attacks. That is what is taking place in the innate immunity. You do not have any kind of memory that is taking place, whereas your memory cells will be activated in the adaptive immunity. Again, you can just see the different kinds of immune system cells, myeloid, lymphoid. Myeloid has granulocytic, granulocytic has neutrophils, basophils, eosinophils. Monocytic has macrophages, Kupffer cells, dendritic cells. Lymphoid cells are again T, B, and natural killer cells. TS, again you have helper T, suppressor T, and cytotoxic T cells. B cells are again memory cells and plasma cells that are present. So like I told you, these are the neutrophils, monophysites, and macrophages that are present. And these are the natural killer cells and the eosinophils that are present. So lymphoid cells, 20 to 40% of the body cells are leukocytes, in fact. And all of them mostly are from the lymph nodes. They are, if they are present in the naive form or inactivated form, they are known as naive cells. And nucleus almost is very big. entire cell is being occupied by the nucleus and it is almost 6 micrometers in diameter. I've already dealt with in detail just a little while ago, so I'm not going to go into details. You can just see stem cells, lymphoid and myeloid precursors, which gives rise to different kinds of lymphocytes and different kinds of granulocytes that are present. Please remember this particular slide because it is very important. How do you identify them? Because you have the cluster of differentiation. So you have around 300 cluster of differentiation of CD markers that are present, especially for T cells, that is the cytotoxic cells. You have the CD4, CD8 and the CD3 markers. For B cells, you have the CD19 marker. For natural killer cells, you have CD56 marker. And the dendritic cells, all of it have different kinds of CD markers present on the on the cell surface. Mononuclear cells, again based on the tissue that they're present, we have already seen on the particular side, and how the action of the mononuclear phagocyte takes place. Dendritic cell also action, and it is using your it is help to the tumor treatment basically. And mechanical factors are here, different kinds of epithelium, because and mechanism is desquamation or peristalsis or flushing action that is taking place. System or organ that is given. Numerous components are these. Again, what are they? Lysozyme breaks down the bacterial cell wall, lactoferrin transferrin, complete bacterium for iron. Cytokines have different kinds of effects. Again, complementary system helps in removing or lysozymal activity, phagocytosis of the bacterial cell that takes place. So different kinds of cells have different kinds of activities that are present. So we have just seen this again, non-specific kind of immunity that is present. So I'll let you know that's what. Adaptive immunity has again different kinds, again attributes that are present. Antibody specificity, diversity, memory cells, and self and non-self recognition cells that are place. And it distinguishes minute differences that are present basically. And depending upon the kind of antigen that is there, it will adapt itself. Have memory cells which help in recognizing the antigens that are present. And you have non-self antigens which recognizes the different kinds of antigens. Again, and you have the humoral versus cell-mediated immune response, wherein humoral immune response, what is happening is antigen gets coated or gets attached to the antibody, because of which antibody gets destroyed. So there's a lot of cross-linking takes place, clumping formation takes place. Cell-mediated, what is happening is first T helper cells come, activate the phagocytic cells, then this activates your B cells, then the antibodies are produced. Then what will happen is then finally clumping and such kind of again the secondary response is going to take place. And then we have the activated T cells which kills the altered cells or the viral infected cells that are present. So these are the T and the B lymphocytes in their respective functions. Cytotoxic T cells provide the cell-mediated immunity. In fact, helper T cells help in stimulating other immune cells and production of cytokines. Other memory cells will jumpstart the immune system upon reinfection. So we have CD4 and CD8 cells and the MHC complexes 1 and 2 which come and take part. And then we have something called as the antibodies that are going to be there. We will read about it in some time. This is the basics of your immune system. Now I want to talk about basic antigens. Antigens are those which help in please give me a moment. We have antigens right now, so I will list it. So when it comes to the definition of antigens, we see that antigens have different kinds. They are known as your haptens, epitopes, or antigenic determinants and antibodies that are present basically. Just So, what are antigens? Substances that are capable of inducing a specific immune response. They will recognize the surface antibody cells or the T cells receptors that are associated with your MHC molecules, and they bind specifically with an antibody or T cell receptor. And you have different kinds of antigens that are present. Basically, you have exogenous, endogenous antigens, and endogenous antigens that are present as xenogenic, allogenic, or autologous. So, what does it exactly mean? Exogenous that enter the host from the surroundings or external environments. So, your drugs, your pollen grains, your peanut. Some people have peanut allergy, and some people are allergic to brinjal. When they consume brinjal, they start getting an allergy. So, they're all exogenous antigens that are present. Now, what are endogenous antigens? They are either xenogenic or heterogenic, allogenic or idiotypic, or autologous antigens. So when it comes to the definitions, when see, xenogenic antigens are those which are present, especially when somebody undergoes a surgery for grafting. If there is any kind of kidney transplant, liver transplant, there is antigens produced against, the new liver or the new kidney is treated as a foreign entity. That is when antibodies are produced against them. Such kind of substances are they're known as your xenogenic antigens. Allogenic antigens, foreign items, enter when the individual undergoes a blood transfusion or there's any kind of transplantation again. And when there is autologous antigens, it's rare and unnatural in a normal condition basically. So it is self in nature and normal in fact again. So what is exactly immunogenicity and antigenicity? So whenever there is only a or self-mediated immune response happening, then it is known as humoral or immunogenicity. If you have completely production of different kinds of antibodies taking place along with complementary system or something called as opsonization taking place, that is known as antigenicity. So anything can be immunogen, but anything can be an antigen, but it should trigger the immune response within yourself, within the body, so that it produces the different kinds of antibodies. That is when it is known as your antigenicity or immunogenicity. All the molecules that have the property of immunogenicity are also having the property of antigenicity. Meaning antigen can be anything, but if it is an immunogen, it can be called as an immunogen only when it triggers an immune system reaction. Okay, so some molecules have only antigenicity, but they do not have the property for or trigger the immune response in our body. Such kind of substances are known as haptens. You see here, amino acids are over here, haptens, lipids, steroids, carbohydrates, and proteins. So this is how an immune system will be triggered or increased. This will not cause an immune reaction to take place. But when it becomes a complete carbohydrate or a protein, then only the immune system is going to be triggered. So what are the factors that will influence the immunogenicity? It is how much amount of the immunogen is present. How foreign is it? What is the molecular size of it, the chemical composition, the physical form? Is it going to be easily degraded once it gets into the system? And what are the different kinds of factors of the biological system also? Are we genetically prone to or susceptible to different kinds of diseases? What is our age? That matters. What is the method of administration? Is it a dosage that you're taking in, or is it a root the skin, or is it an adjuvant that is given will increase the immunogenicity factor also. That is why it is when you're giving an a vaccine, you have a hapten and you have an adjuvant attached so that it will help in increasing because the molecular weight increases, it will increase, or for sure you have the production of the immunogenicity taking place. So, like I told you, immune system discrimination between self and non-self. Only foreign molecules are immunogenic when an antigen is introduced to the organism. molecular size. The larger the molecule, the more immunogenic it is. So difference between 100,000 Dalton to 5 to 10,000 Dalton makes a difference. Of course, you can just see 100 angstroms, glucose, and this particular, what do you call it, chart has been given. Chemical composition, more amount of aromatic amino acids such as tyrosine or phenylalanine can have a profound effect, meaning it will definitely trigger your immune system. Physical form, those which are insoluble in nature that as compared to soluble or denatured ones are more immunogenic. Degradability also, those that are easily phagocytosis are generally more immunogenic. And they should be able to be phagocytosed, processed, presented to your antigen presenting cells to the T helper cells that are present. Of course, genetics plays a very important role because the person having an altered genetic code, as it takes place in the case of certain people who have the recessive or mutated genes, will definitely not be having appropriate genes for the APC cells to be produced. So, because of which the immunogen goes undetected. Age, very young people do not have such kind of matured cells that are present. Very old people are not able to have a good, proper immune system that is present. That is why they are the most vulnerable and susceptible when it comes to anything. Dosage also. Small dose might not have an impact, but the larger the doses, it will have a definite impact on the immune system for it to be triggered. Excessively high dose, again, it is going to be fatal to the organism. Adjuvants are those which will enhance the immunogenicity of the antigen that is taking place. So what is happening is with an antigen only, you will have time for the antibody concentration to be there. But if there is antigen plus adjuvant, your concentration of the antibody will definitely increase. And the time also is going to be there. So how much amount of adjuvant that has to be given should be taken care of so that you have maximum immunological response being created in the body. And you have your alum, common alum, potassium aluminium sulfate increases the antigen persistence. So this antigen mixed with alum salt precipitates the injection. Slowly this will go inside and it will take slowly for the cells to come, be exposed to the particular antigens, and then you have the memory cells that are going to be triggered. Right. So you have even France water in adjuvants basically that is present. Water in oil adjuvants and incomplete and complete adjuvants that are present basically. So incomplete ones are when you use your mineral oil, emulsifying agents such as mannite, manoliate and all that. In fact, and complete adjuvant is, for example, your heat-killed mycobacterium, tuberculosis bacteria in the water and oil emulsion. So you add around protein, which is muramyl dipeptide, which is there in the bacterial cell wall. This activates the macrophages. So these activated macrophages are definitely going to have a very good, what do you call it, system basically of immunogenic to trigger the immune response. Epitopes, the part in the antigen that is going to be recognized by the immune system or the antibody is known as the epitope. Usually protein part, and that is there in the case of protein antigens. So it has primary, secondary, tertiary structures that are involved. And in the case of different kinds of antigens that are not proteinaceous in nature, you have glycosidic bonds and 3D conformations that are present. Can be of anything, but usually proteinaceous in nature. These are going to be recognized by the antibodies that are present, and then TNC cells exhibit these fundamental differences in the recognition. So these are the antibodies. In fact, this is the antigen of the bacterium, epitope region. Then this is the hypotope region, and then this comes over here, and then this is the B cell. After B cell, it is going to be taken down to MHC complex, MHC class 2 cells. B cells, and this is how it is going to be recognized by others. Now we have something called as the mitogens. They're agents capable of inducing cell division at a high percentage in both T or B cells. So mitogen can activate many forms of your B cells basically. So that is why they are known as mitogens or polyclonal activators. So what are they? Now you have lectin molecules that bind to membrane glycoproteins. So this leads to agglutination or clumping. And then you have some mitogens that activate your B cells and some activate the T cells. So three common lectins are present, which are concavenin A, phytohemagglutinin A, or PHA as it is known as, and covalent mitogen, which is CW. So these are proteins basically that are very important for us in the biological system basically. And if you see the lipopolysaccharide component of the different kinds of bacterial cells, especially the gram-negative bacteria, you have B cell mitogen. So this LPS, that is your lipopolysaccharide layer, has a mitogenic activity because of the presence of the lipids in them. So that interacts with the plasma membrane. This causes the cellular signal to be activated. Next, super antigens. Very important T cell mitogens are present. They bind to the residues in the variable domain of the T cell receptor and to the class 2 major histocompatibility molecules outside the antigen antibody cleft. So, what will happen is you have a cross linking that is taking place with a T cell to a class 2 MHC molecule, even when the T cell that is, will not be able to recognize the antigen present. So, what happens is it leads to the activation of the T cells. Thus, super antigens can activate all the T cells and they expressing the V domain that is present, which is where the super antigen binds. V stands for variable as such. We have very few or we have quite a number of antigens that are present, which are all super antigens. Example is Staphylococcus enterotoxins, toxic shock syndrome toxin, that is TSS1, that is produced by your gram-positive bacteria such as Staph aureus. So this is about your antigens such as this. Now I will teach about the antigen. We have just we'll have a look at it right now. We have just seen antigens, actins, and what they are. So an antigen antibody binding is reversible also. And then epitope is the site wherein which is recognized by the antibody. So not 1 to 6 or amino acids that are present are going to give their specific antigen. So uniqueness depends upon different kinds of binding sites that are present based on the types of antibodies that are present. So you have an antigenic determinant, which is a cluster of epitopes on the surface of the antigen. So you can just see one antigen, two antigenic, many antigenic that are present. This is where your antibody comes in, starts binding. So this is how an antibody looks like, antigen antibodies. So this is the antigen binding site that is inside amino site with fat. This is the carboxy terminal. We are talking about a protein over here because all your antibodies are proteins in nature. This is the hinge region having a disulfide bond. This is how the basic antibody looks like. So now we have something as the avidity. So avidity is the strength of binding two molecules or cells to one another at multiple sites. It is determined by your heterogeneity of the antibodies in the serum and heterogeneity in the antigenic of the antigenic determinant. It is also known as the Bohr effect, which is K avid, which is equal to K1 into K2. So that's what. And So we have avidity, which is measure of the functional affinity of an antiserum for the whole antigen that is taking place. And specificity is when the antibody is not going to be absolute. You have a cross-reactivity also taking place due to the presence of an antiserum reacting partially related antigen. That monoclonal antibodies also, and this is directed towards a single epitope, and this is because of the cross-reactivity that is taking place. So what are the factors? Of course, like I already told you, we have different kinds of factors that are going to be contributing to the immunogen structure, which is your foreignness, the size, and of course your primary, secondary, tertiary, and the quaternary structures that make an impact, and that will definitely cause that is your combinational determinants. Immunogenicity is also there. Basically, like I told you, foreignness, chemical composition, degradability. We have just seen this, and of course, biological system, age, dose, route, adjuvant nature. Chemical nature being proteins, polysaccharides. Proteins will definitely trigger your immune system, and of course, anything that is foreign is going to trigger the immune system as such. Yes. Types of antigens, T-dependent and T-independent will be there. T-independent are your polysaccharides. Type 1 and type 2 are there. They are going to be polyclonal reactivation that is taking place. Example is a pneumococcal polysaccharide, glycopolysaccharide, LPS, the flagella. Antigen dependent or T-dependent antigens are your proteins. Example is your microbial proteins and non and altered cell proteins. And now we have something called as the immunoglobulins, which are proteinaceous in nature. We will just have to come to it in some time, basically. And fragments, basically, helper T cells and cytotoxic C cells with different kinds of major histocompatibility complex will recognize your antigens and trigger the antibody reaction. So based on the composition, the size, and the number that is present, this is how a conventional antigen looks like. And this is your immune system that is present. APC is present here. MHC class 2 is present. T cell receptor, T lymphocytes. You have the antigen over here. A super antigen has variable sites being recognized, like I already told you, with carbohydrates, T lymphocytes, and MHC class 2 molecules, and the T receptors again. So this is how. So super antigens are produced by pathogens. So they are not able to be processed by the APCs that are present. Example is your enterotoxin, pyrogenic toxins. Your APC cells, like I told you, have the capacity to capture. They are your antigens or APCs. They can activate a non-native T cell that is present. Pathway of presenting is also been here. We can just see this when we read about it. So we have CD4 plus T cells that come. CD4 marker which is present, that is a glycoprotein. 70% of the T cells that are present, they are the T helper cells. They help in modulating the cellular secretion of cytokines. So B cells are activated, immunoglobulins are secreted, macrophages and dendritic cells are activated, and then you have inflammatory pathway. So you have T helper 1, T helper 2. Different types of cytokines are produced, different types of interferons are produced in TH1. TH2 has interleukins that are used. They help in providing the B cells in antibody response, whereas over here macrophages and B cells, that is your dendritic cells, are produced. And you have your cellular mediated inflammatory pathway that is being triggered over here. Whereas over here you have your allergy to extracellular pathogens being used. So this is how the T cells are present. We will just look into it in next class. In fact, now we have something called as your in the antigens itself. APCs, like I told you, antigen presenting cells. That's been there. Differences over T cells are present again. CD4 and cytotoxic cells, T humoral cells, and your T suppressor cells are present. Now, helper T cells include yours have the glycoprotein CD4 and CD8. And they are cytotoxic in nature, and they are involved in antibody production, recognition of different kinds of exogenous antigen that is present, and they recognize your major histocompatibility complex class 2 molecules. So example is macrophages, dendritic cells, and B cells. CD8 recognize the endogenous antigen, so especially the viruses and the cancer cells recognize the MHC class 1 and they are present on all the cells. So in diseases such as HIV, your CD4 and CD8 cells are being destroyed, because of which what will happen is you are not able to catch hold of the virus, and the antigen presenting cell is not able to come and identify the virus in the first place. So because of which you have a problem. So this is how the antigen recognition takes place. Here, this is the antibody structure as such, basic antibody structure. You have the complementary binding site, phagocyte receptor binding site. This is how an antibody in IgG looks like. So TCR recognizes its epitope. So we have two types of epitope: CD4, MHC class 2, CD8, MHC class 1. So MHC class 2 antigen, that is your TCR, CD4 cells that are present, CD4 T cells. So this is how MHC class 1 and 2 cells are present on the APC cells, that is they bind only to the smaller peptides and they are not generated by any kind of rearrangements. So they're important in the transplantation. They're known as a major histocompatibility complex. T cytotoxic cells, like I told you, have CD8 molecules on the surface. That 30% of the cells, they destroy the cells that are cancer cells and your intracellular pathogens. They release the perforins and the granzymes that are present over here. This causes the cellular lysis and apoptosis. And when there is a humoral response that is being activated, what is going to happen is you will have helper T cells that are being stimulated by the T-dependent antigens. Then this will release the ABCs along with your class 2 MHC molecules. And then your helper T cells get activated. So class 2 protein is present. And then to that, you have your bacterial antigen fragments. This is when your CD4 cells come into place, and then, with this particular complex being formed, other cytokines and interleukins are going to come into action, and this causes the antigen to be fragmented. And what is going to happen is your complete, immune responses will be triggered. And so this is what is important. What we have to see is binding of antibodies to antigens. So viruses and bacteria, what is happening is neutralization. When it is bacteria, you're going to have something called as agglutination. And then you have something called as a precipitation in the case of the antigens if they are soluble in nature. Or else we have a complementary fixation wherein a complement comes, holds the antigen to the antibody, and then this causes other kind of complement reactions. All this finally, what is going to happen in the case of neutralization, agglutination, and precipitation, you have phagocytosis, wherein a macrophage comes, takes in that particular antigen, is going to be destroyed, and the products are the antigen fragment products are going to be released. Whereas in the case of complement fixation, this leads to the cell to completely die. So the cell is also dying in the first place. So we have to first see what is important is location of the pathogen. Are there viruses in the cytoplasm of the cell? And how is the MHC class 1 pathway important? Are we having the cytotoxic T cell response? And then what are the different kinds of bacteria which are present extracellularly inside the lymphatic system or inside the vascular system, outside the cell? What is the pathway of the MHC class 2? And then how are the T helper type 2 cells are responding? And what is the antibody formation? So we just have to look into it. This is all for today, basically. Thank you so much.
Excuse me, ma'am.
Yes, ma'am.
Ma'am, in bone marrow, hematopoietic cells grows on the mature meshwork of lymphocytes, ma'am.
In the bone marrow, maturation of the lymphocytes will not take place. The maturation of the lymphocytes will take place in the secondary lymphoid organs. Bone marrow produces the lymphocytes. Maturation takes place in the secondary lymphoid organs. That is your thymus, that is your spleen, and other places.
Okay.
Is it clear?
Antigen
Right,
cells.
Yes, ma'am.
What are antigen presenting cells, ma'am?
Antigen presenting cells are those cells which help in recognizing the antigen for what it is, and then it goes and presents the cells to the different kinds of lymphocytes, to class 2 proteins, that is your MHC proteins, and then to the helper T cells. So your T cells are of three types. T helper cells will be there, T suppressor cells will be there, T cytotoxic T cells will be there, depending upon the function. So antigen presenting cells will take the antigen and present it or get attached it to the T cells. So this in turn what will happen is secrete the cytokines that will activate the cytokine pathway, and then at the same time it activates the B lymphocytes. The B cells are the plasma cells and the memory cells. So the memory cells will help in remembering the particular antigen for what it is. The plasma cells will produce the antibodies also. So the B cells will get differentiated into the antibodies now. Is it clear?
Okay.
All right. Thank you
Thank
so
you.
much. Yeah. We'll meet up tomorrow. Thank you. How do I leave it? Stop sharing.